back up a little bit here for you. As long as our DVL keeps looking good. I'm going to work further upslope over here.
Yeah, I mean, that way you can like stay in the middle.
Keep going across, yeah. Yeah, I agree.
So this dive is anticipated to be a 12-hour dive, and we'll likely be heading back up around 9 or 10 Pacific time. So there's a lot to see in the next few hours.
Are these anemones?
Yes,
Right?
with the
They're
really long
so
tentacles.
different. Yeah.
And then there are corals on stalks with 3 fronds. Those are a type of black coral.
I feel like the tentacles on some of the anemones are longer. Is that necessary at greater depths to just meet the nutrition or energy requirements and capture more food?
Yeah, it's possible that it's an adaptation to increase their chances of capturing marine snow, which
Hang out
is the
here.
food at this depth. But these anemones definitely have longer tentacles than I'm used to seeing them with.
Roger that.
And can you guys remind us what other sensors and measurements are we taking when we're at these depths?
So on the ROV, there's a CTD, which stands for conductivity, temperature, and depth. And there's also an oxygen sensor. The water samples we collect will be analyzed for carbonate chemistry, as well as environmental DNA. The sediment cores will be analyzed for microfossils, which are forams. I'm trying to think.
Are we collecting any rocks?
That is not a priority for us on this mission. Nav, what is the distance from where we are to that point 38? I know we haven't made much progress.
We're not doing bad.
Okay.
Let me check. I'll set the target to that.
Okay.
And at over 3,000 meters, it's pretty cold, so just a little bit above freezing. We're seeing water temperatures at about 1.6 degrees Celsius, which is colder than we've seen throughout our dives.
Another step, please. How did we get in feet? What happened?
Feet.
Yeah. That's what I said. And why is my target 050?
What a total Shih Tzu dog.
Oh, I wanted to have a bow on the top.
Maybe you want to go ahead and push it on the dog?
It's a pom-pom anemone.
Mike, could you able to come down a little bit on the winch?
Yeah.
Liptonema.
Thanks.
Thank you. I got some really good photos.
Roger. So about the dog.
Oh, huh?
Give it a scratch on the head. Under the chin. What is this?
Check that out. Oh, a rock.
Oh, yeah, that was eventful. Okay, go ahead and come wide.
Not interesting enough.
Rocks are cool, but there's squishy things here for us to look at.
Can we look at the sea star above the lasers?
Yeah, of course.
So the vehicles are about 370 meters off of 38.
Okay.
It's also kind of into this slope, so I'm not sure if they'll actually go to that spot. It's a bit sketchy.
Okay.
Bridge nav.
Thank you.
Another step, please.
That's good. Thank you.
Right.
There's the sea
A sea
pig.
pig.
What? Where?
That clear thing.
There it
like,
is.
How about we
Like
look at it?
translucent
Let's look
kind
at
of
it.
thing.
So this is a sea cucumber.
Wow.
This is my Halloween scary.
Someone needs to make a costume.
Yeah.
Like this.
Yeah.
Are you saying Sam Wishnack needs to make a sea pig costume?
It's green up to
So
say
yeah,
hello.
it's snacking.
Wow.
So weird.
All right, go ahead and come wide there, video.
That is
It's
my
really cool.
first.
Like sometimes you can like, depending on the angle you're looking at, you can see all the insides.
Oh,
Yeah, like the
interesting.
camera is just such a good camera. You can.
It's a very colorful squat lobster. Squat lobster?
Yeah, I'd call it that.
Looks like it, yeah.
Yeah.
Is that one falling?
That's one floating down.
Yeah.
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 Are these are corals, the same black
Yes,
corals?
these are black corals. You're right. They're quite common.
Oh wow.
These little pockets of life are so cool.
Yeah.
Yeah.
Oh, there's a sea spider hanging off of that.
Oh, is that what
Oh,
it is? I
yeah.
saw
Do you guys
that.
want to look?
Yeah, let's look at that.
All right, video, go ahead and start pushing. That's good.
That's neat.
Oh, wow, that's cool.
Yeah.
All right, Miles is back. See you
Is that-
guys later.
Bye,
Thanks, Erin.
Erin.
Thanks, Aaron.
There you go. I got some good pictures. Ah, there. Perfect.
All right, video, go ahead and come wide, please. Pretty floaty.
One of our viewers is asking about how much of the ocean floor has actually been explored, and is it true that we've seen more of the moon and Mars than we have our own ocean?
Yes, I think that's an accurate
Yeah.
statement.
Yeah. Actually, I was trying to show my roommate Joe last night a TED talk with Dr. Ballard, and I think he talks about that actually. So if you're into TED Talks, you can go on to TED and look up Dr. Bob Ballard, and he's got a great talk there.
Hi, Danny is back in the lead watch seat.
Hello, hello.
Hello.
To the viewer
Hi,
who's
Hugh.
asking
Would you
you,
mind coming down a little bit there?
Do we know how tall this feature is?
Miles?
Not off the top of my head.
Okay.
Looks like a little more than 100 meters before it would sort of initially crest.
Wow.
we have seen debris on the ocean floor throughout our diving here in the marine sanctuaries. This is a question from one of our viewers.
Can I get a zoom and some photos of that grenadier?
There we go. You can get the whole fish.
Nice.
Hey, I got some images.
That's my thought. But I mean, somebody could do that if she gets zooms and it's all good, then it's like whatever.
It's incredible. We're at 3,300 meters and to see fish at this depth.
Yeah.
Really good, and then whenever I get a little break, like if they're not on me for something, I'll let it drip for a minute, and even if it's almost perfect.
All right, I heard there was a question about mapping the oceans, or ocean. We have one ocean, several ocean basins, and that is, there is a goal, Seabed 2030, to map the ocean floor at the best possible resolution. However, some of the ocean floor is super deep, which makes getting high resolution data of that seafloor really difficult. So Sage pulled up a site for me, Seabed 2030, that shares out what the plans are, and 0 to 1,500 meters. Percent of the world ocean floor is 13.7. Okay, now what's it? The table shows the minimum resolutions we expect to achieve at each depth range by 2030. So 13.7% in the 0 to 1,500 meter range. 1,500 to 3,000 meter range, 11%. And 3,000 to 5,750 meters, 72.6. That's pretty cool.
Yeah.
That's like at the depths we're at now. We're at 3,308 meters right now, which means 72.6% of the world ocean floor at this depth and deeper to 5,750 will be mapped by 2030. And then the deeper, 5,750 to 1,100 meters, 2.7%. So that is very exciting to hear about.
Yeah, I think we moved stuff around a bit, but everybody, their targets are pointed south.
This is an international network of leading ocean scientists and ocean mapping experts. The Nippon Foundation Gebco Seabed 2030 project.
Sorry, I tried to catch them as they were going out the door, leaving stuff all over the control console. So we're lateraling left. Easy, easy. Well, they did switch it up. Look, they drew a wave on the nav screen.
So interesting, spoonworms are black down here. We've been seeing white ones.
That's what those are. Okay. I thought they were stalks from something.
So we saw them on earlier dives. When you zoom in and spend some time, they slowly pull themselves back in.
Yeah.
All right, here's some rock. Let's see what we find. Let's get a zoom on that white sponge to the left of the anemone.
Is that good, or do you want it zoomed in?
I think that's pretty good.
Okay, I got some few
You
images.
can zoom in a little more if you want, Wayne.
Yeah, we just got to this rock. We can zoom out.
Okay, thank you. Got some good pictures.
There's another Bathypathes at the bottom of the screen. They look pretty big down here, and they're yellow, which we haven't really seen before. We mostly see them pinkish, orangish, or even red.
So to our viewers tuning in, asking about what depth that we're at, we're currently about, I think just over 3,200 meters, which is 10,850 feet.
That's great, thank you.
To calculate the pressure, what you would do is you would divide 10,850 by 30, and then multiply that number by 14.7. That will give you the pressure in pound per square inch at this depth, which is probably around 5,000 PSI, maybe a little bit more than that.
Our Herc depth is reading 3,310 meters, and just above 0 degrees Celsius.
Wow.
What's the difference between temperature?
Are you guys happy with that shot? We can
Yes,
move on?
happy with that.
We've got two readings on our data here. Temperature degrees Celsius and temperature probe degrees Celsius. What's the difference?
The probe is, sometimes on the Herc we have a temperature probe that we can stick into a sediment or methane seep or hydrothermal vent, and then get a temperature for that. So
cool.
So which is the better one to look at? Because I see they look, well, no, they do look.
I don't know. Have you used a probe?
No.
No.
No, but we're almost at 1.5 degrees Celsius, which is colder than where we were the other day. It was like 2
Yeah, it's about
degrees.
two something.
So Danny, we've got a new viewer who just tuned in, and he's curious what are we hoping to accomplish on this dive?
Okay, so we are in Bodega Canyon in Cordell Bank National Marine Sanctuary. This is the deepest depth we've ever explored, 3,300 meters. And this is really an exploratory dive for us. We want to see what the community looks like in these deep habitats and what the substrate looks like along this canyon. The goal is to inventory the species in the sanctuary. We don't even know, because we've never been here, we don't even know what is here. And we want to look at how the communities change at these different depth zones. So we have explored Bodega Canyon a little bit shallower on a previous mission here in 2017. And this is a revisit to look at some of the areas that we didn't get to on those dives.
Great. And do you think
Can we zoom in at the bottom of that rock, just to the right of the lasers? Oh, I guess it's a fly trap anemone. Okay, I can zoom out. Oh, and what about to the just to the right of the lasers, and there's like a white, I don't know if it's a coral or a sponge, or maybe it's part of the rock. Actually, we're a little far away now, so I can't really tell. Yeah, just above the lasers there.
Sorry, I think we're getting a bit of the telegraph from the bridge. I mean from Argus.
10 meters due south.
Is that a bamboo coral?
It looks like a
we'll
bamboo
slide
coral.
A little closer.
It's like a little one below it too.
It's pretty small. It just looks like a little over 10, maybe 20 centimeters. Oh, and there's, yeah, that little one. I didn't see it first.
I got a few images.
Did you say we're at the end of the tether?
We are at the end of the tether, yeah, that's why we're bouncing a bit.
Okay.
We're correcting that now.
Oh, okay.
Put in a
so we're gonna get close, be able to get closer.
Yeah, roger.
What do you guys think, Caitlin and Gary? We'll try and get some closer images of it so you can look at the branching pattern and where the nodes are.
I'll zoom out until we get situated.
So viewers asking about wind at the surface. Why is it that we monitor that? Does it
So I'm
affect?
pulling Argus there. It's not going to be the most stable shot.
Okay, I'm ready whenever we're zoomed in and focused.
Wind at the surface can really affect how the boat moves. And also, we have to keep an eye on the wind to see if it's coming up or coming down. It affects how we can recover or when we need to recover the ROV, because if the wind is really high, it makes recovering the ROV tricky.
Thanks.
The one that we're on right now? Or the one below it? Yeah. I think it's a bamboo coral, and if so, it would be our deepest record. But we're going to get a closer look.
Yeah, we're moving the ship all the time, still bouncing around a bit.
Oh, okay.
Magda, can you give me a rundown of how much space we have for samples left?
Yeah, yeah, we have plenty of room. We've got 4 small bio boxes on the starboard side, and we've got 4 empty slurp jars. And we do have some room in the larger bio box, but it would depend if we can mix 2 samples together. So I think in the past we've done like coral and sponge in the same box, but not like 2 corals or 2 sponges in the same box.
Okay, great. Thank you.
I'm going to be typing. Oh.
Zoom out, Wayne, will you, please? Making me dizzy.
Science team, do you know if there's a website where seafloor mapping data is archived or kept that's available to the public?
NCEI or GMRT.
Oh, thanks.
Sorry? I can't hear you. It's NCEI, NOAA, NCEI, and GMRT.org is sort of a synthesis of sort of all bathymetry and satellite altimetry and everything. That's part of the. They're one of the big collaborators with the JEPCO 2030 initiative, Seabed 2030.
Okay, great. Thank you.
You should have the tether to turn around. All right, I'll give you a little more.
Danny, is Bodega Canyon the deepest canyon in Cordell Bank National Marine Sanctuary?
It's pretty much the only complete canyon that we have in the sanctuary. We have some headless canyons and we have some deep continental slope, but it's the only canyon that goes up onto the continental shelf. Is that right, Guy?
That's right.
I think those are both the same. Okay. Can you find the depth range of the lower? I'm not exactly sure what that is. Viewers were interested in how long it takes for corals such as bamboo corals to get to the size that we're typically seeing.
Well, they're pretty slow growing. All these corals are facing pretty harsh conditions, cold water. They're completely dependent on marine snow that rains down.
Can we hang here for a minute?
And like bamboo corals grow maybe 0.2 centimeters.
Yeah, we're hanging.
Maybe.
So I found that the depth range could be to 2,600 meters, so it's up to you if you want to grab it or not.
All these species are very slow growing. So we know bamboo corals live to at least 400
Okay,
years.
that sounds good.
Not
The
so
reason I
sure
thought
about
it was
other
relevant is
species
the
though.
description has it as being zipper-like polyp in some branches, so you can kind of see that further in here.
Yeah, let's get some close-ups of the polyps. All right, let's try. If you could zoom in on both of these species and get some close-ups on the polyps, that would help.
I'll have to nudge into the wall to get a stable shot, which will probably rain on us. At that level of zoom. Let me swing around and touch there. Yeah, please.
Do you want to get closer to see the polyps, or is that good, Danny?
Stand by for a second. Let me, we'll just touch the bumper bar there and then you'll get a stable zoom.
Great.
That sounds good.
Thanks.
Do you want me to zoom out in the meantime?
Sure.
Danny, can you remind us how fast these corals
Okay,
grow?
Ashley, you can zoom in there.
Are you asking how fast corals grow?
Okay, full zoom there, Ashley.
Yes, that's correct.
Ah.
There we go. Oh, that's a really good image.
Nice.
Is that full zoom, is it?
Yep.
Awesome, thank you.
All right. If we've got some good stills, I think we can move on.
You want to zoom on the other one really quick while we're splatted up against the cliff?
Yeah,
here?
let's do that.
Got little amphipods on them?
Yes. That red thing on the tip is an amphipod, a little crustacean.
Holding on.
Yeah, that's. We've got some very good stills. Thank you.
Okay.
Okay, we can zoom out, keep going.
Okay. Sorry that took so long.
So you don't think those are bamboo corals?
No, I thought when I saw them from a distance, I thought maybe they were because of the shape, but I don't think. I think maybe. I don't think it is now. It's another one of those kind of blobby things right there. Can you get a zoom on that? Yeah, the quick one is good. We saw that earlier, and I don't know what that is.
Wonder if it's attached to that thing, or is it
think it
an
is.
extension of it?
Okay, we can zoom out and keep going.
Sorry guys, we're up against a cliff there and I gotta thrust back to get off it.
Yeah, that's all right. Corals are really slow growing and can be long-lived species, so we don't know how fast some of them grow. One of the facts that we've seen recently about, oh, what is that stick kind of thing? Can I get a zoom on that? Whoa.
Cousin it.
What is, could that be a rock pen? Look at that base. No, I guess not.
So some serious polyps.
Yeah, what is that? Could those be zoanthids on something?
There's a bunch of stuff inside there.
Yeah.
There's like this filament, dark. At the base, it's black.
I know. Oh, that's what I was thinking, that those could be zoanthids, and look at the base, that it's black.
Interesting.
Because those don't look, doesn't look like octocoral polyp.
There's a lot of tentacles.
Yeah. So, an idea that that's a black coral with zoanthids growing on it.
Do you think the coral at this rate will it die? Would it
I think essentially the coral live material is gone, and it's essentially an axis with other animals growing on top of it.
Okay.
And what are some stresses that corals face at this depth?
Does Gary want this? Otherwise, I think I'm going to move on. Okay. All right, we'll move on then.
Copy, and moving on.
Yeah. Question was, what are stresses that corals face?
Yeah, what are stressors? I think we're all familiar with stressors in more coastal or shallower corals, but deep ocean corals, what are some stressors?
Well, one thing that we're concerned about is destruction of deep sea habitat. So that's one reason why we're trying to find out as much as we can about this type of habitat. So if things are proposed that could affect deep sea coral habitat here, or in other places we have more information about the communities that live there. For example, types of fishing or oil and gas exploration or
cable laying,
things like that. Some of those things are not allowed in national marine sanctuaries currently, but could be proposed elsewhere in the future. Also, climate change is a big one. So in the deep sea habitat, a lot of the conditions are fairly constant. It's cold, the dissolved oxygen is fairly constant, so it might not have the kind of stressors of warming ocean temperatures that the surface waters face, but the deep sea is connected to the surface through the food that rains down from the surface. So if conditions change those surface waters, that could definitely change the food sources for these animals. So let's get a zoom on these. The sponge and coral at the bottom here.
Also the chemistry of the water that's
you can zoom in
down here. And that is one of the things that we are taking some water samples to get carbonate chemistry analysis, so we can better understand how carbonate chemistry is down at these depths. And the reason we're concerned for that is that with the burning of fossil fuels and deforestation, we've been putting so much carbon dioxide into the atmosphere, and the ocean has been absorbing it like a sponge. It's actually starting to change the chemistry of the ocean and causing what's called ocean acidification. And animals that need calcium carbonate in the water to build their skeletons, such as deep sea corals and other shelled animals, they're having a harder time getting those carbonates with ocean acidification happening. And it's very different all around the world ocean in terms of the conditions. Some areas are highly acidic.
If we swing to the side a little bit, could we look at that from the side and also the coral that's below it? I'm thinking that this probably might have a stalk on it as well.
Yeah, we'll look at the one below, and then I'll try and come around to the side without
that sounds good.
stirring it up off the cliff there.
So regarding ocean acidification, some areas are having much more corrosive conditions than others. And because of the way the water moves all around the planet, we're interested to see how carbonate chemistry is being changed in the deep sea. So that's one thing we are studying, bringing some lab samples back to Carina Fish at Bodega Marine Lab.
This looks like Lepidopsis. Although it's hard to see the nodes. Oh, it does have a branch.
That's a really good zoom.
Wow, look at those tentacles.
It also, yeah. Oh. There's a branch, and I think I see nodes. I think there might be some anemones on this. Or zoanthids. Can you see that some of those look different? Some of the polyps look more octocorally, and the other ones look like anemones.
Yeah. Mm-hmm. Interesting questions here. How do these zoanthids get here? They just drift and land and decide to start living there? Wow, that's a great zoom.
Well, I think I want to collect this. I would like to collect it below where you saw that short branch coming off. So I'd like to make sure we have that below the branch on there.
Okay.
I'd like to first get some good images so you can see how big the whole animal is. So if we can get a frame with top to bottom on it, that would be good.
This is as wide as I can go.
Okay.
Okay. It's pretty long.
It's climbing onto the vehicle. It wants to come out with us.
I think I got some images of it earlier when we were coming up on the rock.
Great.
Do we get a good image of its base?
It's a good idea.
One of the when we did zoom in initially and started looking at it, I got some images.
Oh, that's good.
I'll give you another one here, but let me turn on a light.
Sounds good.
This is so cool.
Wow, it's really tall. Oh man, I really want to get that branch in, but it's pretty far down.
There we go.
Aaron, Jeremy and Liza from Boston, Massachusetts were curious about our cameras on Herc and kind of their capacity to capture these amazing zooms.
So there's a main camera on both Argus and Herc that are used, and they are HD cameras.
Wow,
And
I don't think I realized how tall this was. Are we going to be able to get this in a box?
We might have to somehow fit it in there. We have some, I mean, we could put it in one of the larger bio boxes with a sponge. I'm just not sure with this length if it will.
It can be, yeah. It's okay if it breaks into pieces, but.
Why are you interested in that branch?
Because the branching patterns on these bamboo corals, that's one thing that we haven't been able to figure out, the different species. And so looking at where it branches has been something we're trying to figure out.
Can you tie it in a knot?
Yeah.
Can you throw it over your shoulders?
Curl it up.
What do you guys think? I could need some collective input here. Where can we put it?
I think it would have to go in a larger bio box.
I think in one of the other. Oh, I can do it. It's
Would it fit in
dance here? Plus. The plus dance.
There are two pairs of cameras that share the same feed.
We do have a sponge in one of the forward bio boxes, a Mega. If we are able to fit it in that one, I think that would be the best.
That would be the best?
Yeah.
Okay, let's do it.
Now you can look at the manip.
Danny, what do you think the length of this coral is?
Oh man. Definitely over a meter. Oh, you can get a good view of that sponge on the stalk back there now too.
Oh yeah.
So this, I believe, is the first, we think this is a bamboo coral, and this is the first one I think we've seen in this dive, so this means that it's our deepest record of bamboo coral in Cornell Bank National Marine Sanctuary.
Somewhere in there?
Yep, that'll work.
Okay, Ashley, if you want to do a full zoom there, I'll position this thing in the...
Hey. I switched with Ashley.
Did you copy that, Ashley?
It's Aaron.
Huh? Aaron, sorry.
We played musical chairs. Aaron's now our video engineer.
Video, can you zoom in, please? Is that full zoom, is it?
No.
Or go for full zoom.
There you go, that's full.
Thank you.
Oh,
I can't see over there.
That's okay. We switched a little while ago. I've been hiding over here.
052.
Oh, that's what you guys are teasing me about.
Yes,
Ah, thanks for cluing me in there.
We're trying to be polite. Like, it's Aaron.
Sorry, I'm looking at coral.
It actually hasn't been that long. I think it's only been like 10 minutes, so it's okay.
Oh, and you were counting. My embarrassment. Thank you.
At least it wasn't like 2 hours later.
It was right around the time everything came in focus.
So you think this is a bamboo?
Yes, I think so. Well, not sure, but that's why we're collecting.
Okay, have you seen enough of this branch yet?
Yes, we're ready to put it in a box.
Roger that.
So
What box would you like it in?
Forward by box Omega.
Well, Aaron, Ashley, whoever's over there now, can you zoom out, please?
So that box already has a sponge in it, but we're gonna
Oh, in with the
double up.
sponge.
Yeah.
Yeah, so
Roger.
just have to watch out. Sponge
Oh, I see.
doesn't float away.
Okay, Argus, you want to slide it out? Now you guys got me all nervous. Going to be formal now.
It's gonna be fine.
Well, yes, Herc.
Oh, that's a really large sponge. They're just gonna have to share.
Yeah, our lab work later is going to be very busy.
Yeah, if anyone wants to help us at midnight.
We'll be there.
Thank
Because
you.
we only have a couple hours to do the work. This cruise ends tomorrow morning.
Should I go for the fold? It's looking like a fold. Let me get this straight. You want this coral and that box, right?
With that sponge, it's okay if it folds
With
up
the
or
sponge.
breaks. Yep.
That's tricky.
Just a few hours left in our dive until the end of the cruise, so we are getting as many samples as we can. It's our last chance. The next watch was telling me when at the dinner break that they're gonna, when they get on watch and want to collect things, they're just gonna start throwing our samples out of the box.
Oh no. Not okay. We just have to come back. We need more time.
Indeed.
It is fun to go to the deepest reaches of the sanctuary. I've been with the sanctuary a long time, and I always have talked about those deep areas, and it's fun to actually be here now to see it.
Oh, I like that technique.
Yeah, I was just thinking like with spaghetti and fork, like you.
Little spiral.
Too bad it's not working.
Need a reel, coral reel.
I don't want to spaghetti the sponge, so. I'm assuming you want the branch not broken, right?
Preferably.
That's the box all the way out, is it?
It is, yes.
I just want to go in there and stuff it in.
Yeah. Feels like you could reach out with your hand.
I'm gonna break it if I do that.
Oh, you're getting close. And although Herc's manipulator arms are pretty amazing at collecting samples, obviously it takes a amazing ROV pilot to also get it into the boxes. It's tricky. Well done.
Good job.
Ah,
Dan.
don't count your
Yeah.
chickens yet.
You're gonna